think you see it all now, is that correct?
Yep.
Great. So in this presentation, I really want to talk about some of the thinking that we've been doing around how we might need to improve how we develop our trials in stroke recovery for the purpose of trying to do better research that can give us some breakthrough treatment. That's our goal. And so I just want to start by really defining what we mean when we talk about rehabilitation. We talk about a process of care or services. And I'm not going to talk so much about trials that look at improving rehabilitation. There's a particular methodology for that. We can discuss that maybe later. But I do want to talk about trials where we're talking about recovery. So interventions where we're aiming to really promote a return towards pre-stroke state. And when we think about these, we're also really interested in not just looking at things that we might be able to do that help people improve by compensation, so by adapting to their stroke, but also, really importantly, things that might enhance biological recovery and really aim at a behavioural restitution level. So where we're really trying to make a difference that isn't about people compensating for their problems after stroke. So one of the things we've contemplated is what is the timing of changes that happen, and a lot of this information is really informed by our pre-clinical colleagues, people who've been studying how the brain repairs after stroke, but also with insights from some of our advanced imaging. So we are really starting to get a sense, or we feel it's important to start to talk about time points or time phases in a consistent manner. So here in this slide, we're talking about hyperacute periods, acute periods, early subacute, late subacute, and chronic. And we're going to try and use these terminology to help us be a bit more consistent, because at the moment, people can talk about acute and they can be talking about a study done at 3 months or a study done in a week. So that's why we're looking at this. And of course, what we see with spontaneous recovery and some of the really useful plasticity that occurs, it does happen early. And so this period is an optimal period for us to think about as we're doing our stroke study. But why haven't we really had any great breakthroughs so far? Well, we think it's probably a combination of perhaps not having the best research designs in how we design and ask our questions and then design research around them. Maybe we're not working at the right time with our studies, so we might be applying our interventions later when in fact they may be better tested in an earlier time frame. And we certainly have done a lot of research where we tend to work in isolation, not collaboratively with preclinical researchers, but also with each other to build good studies. And of course, one of our big challenges is that we really just don't know what is going on, how do we pick our people to target for our treatments, and what are the active ingredients in the treatments that we're aiming for that really are going to help promote recovery. And this isn't simple. So I certainly don't want people to feel that we're failing. We've learnt so much, but it is a very big and complex research question that we have to solve. So I just want to highlight a couple of things that as we look into how our studies are done, we look at what our evidence base is. So this is a study that we did a couple of years ago, and we were looking at the Cochrane reviews of any interventions that weren't drug or device, and what was the backbone of the evidence that we had from these Cochrane reviews, which are important. They help guide out some of our decisions. Well, we'd really like them to if we could get summary evidence that told us that this was a good treatment. So there were 197 trials in this review, and nearly 10,000 patients. But we see some of the problems with not having studies that are between more than one centre, and I'll come back to why that's important. Making sure that we have some rigour in the methodology and the sizes of the studies to be able to show an effect. If we're doing small studies and we're comparing one intervention with another, we really must expect to do a small study. We need to have an intervention that's going to have a powerful effect to be able to see anything. And to be honest, a lot of our interventions might be efficacious, but they're not a really powerful effect. So these are some of the problems. This one's a bit busy, but it comes from another review that came out just last year, done by Meredith Firth, a PhD student that I work with. And this is now looking at drug studies, because when we consider the interventions that might make improvements to recovery, we need to think about other treatments as well, like recovery-promoting drugs. So I just want to point out that when we did this review, which is a really nice summary of these drugs that are aiming at recovery, there are a number of studies, many different agents, lots of different outcomes. Most of the trials are done late, which may be appropriate for some of the interventions. And the paired treatments are poorly defined. So we would hope that for a lot of recovery-promoting drugs, we actually need to drive the system. And driving the system would require some kind of training, and we don't see well-described training paradigms. And again, these studies are quite small. And then it's not just a question of them being small or big, because when we look at the recent large trials that were reported just last year, the fluoxetine for recovery trial, which had over 3,000 patients, and the Rattles trial, which is the robot-assisted training trial for recovery, both very well conducted studies, but they didn't find benefit. Now, their development may have been, the right kind of process, but there's something about how we're doing them perhaps that can be optimised to try and get to interventions that can help people recover. So when we think about these, we're wondering, is it the timing? They were done some early, the focus started early, but the Rattles trial had people from a very wide range of time points after stroke. Is it the target? Is it the dose? Is it the patients that we're putting in? So who are the target group that we really want to have in our study? So we have lots of trials. There's a lot of activity in this space, and some of them very, very good trials. But we think it's time for us to think about how do we make them better, even better. And also, we've got this really growing group of people interested in this space. So how do we also build trial capability so that we've got better trials? So a couple of the things that we've been thinking as an international group is around how we do our trials and develop standards for our trials. How do we collaborate more to build our studies and do more multi-centre trials? And also, how do we encourage rehabilitation professionals, clinicians, scientists to work more closely together as we try and build these recovery-promoting treatments? So I want to tell you about some of the collaborative efforts that have been going now for 4 or 5 years and what we're aiming to do with those, and invite you to participate in the new stages of this activity as well. So in 2016, we ran the first stroke recovery and rehabilitation roundtable, and we did another one in 2018. And this was to look at the gaps and possible solutions to some of the challenges we're facing at the moment in our trials and research space. And I do want to emphasise that all of these papers are available online, free, in the International Journal of Stroke, and also in the MNR. So I just want to highlight from the first roundtable, one of the things that we really wanted to achieve, as well as looking at the issue of biomarkers and how do we find recovery biomarkers, and also development of interventions, I encourage you to look at, is we also talked about how do we standardise assessment in our trials, both when it's done, what is done, and also we came up with core outcome measures that we think should be present in every recovery trial to try and give us more capability later to pull our data and to look at the effects across our research study. But for the second one, which I'll spend a bit more time on, we had one of the groups that worked on how do our trials need to change, and I'll talk more about that. We also had a group looking at cognition, because this is a neglected area in terms of definition, consensus around targets, really complex area. When we think about it, we actually know a lot more about motor recovery than anything else in stroke recovery. And also translating evidence into rehab practice, that's certainly another area. And the other area that is also relevant for recovery trials is how do we measure recovery, not just function. So how do we go beyond looking at a simple tool to measure outcome like Kugelmeyer or the modified Rankin scale, and actually measure something that is trying to tap into biological change and repair. So I'm going to talk about the Recovery Trials Task Force, which Steve Kramer and I led with a fantastic group of both preclinical scientists and clinicians, as well as trials developers and statisticians. And we worked around how do we create a development framework that might help people coming new into doing research think through some of the critical elements that we need to get right if we're going to give ourselves the best chance of getting a good study with a good target for the right patient at the right time. And that's what I'll talk you through. The main thing that we focused on was what we called identifying key knowledge units, which are key things that we need to consider as we're building our trials to be able to clarify what the evidence is and make sure that we've got the best opportunity. So in this example, we're talking about how much intervention might be delivered, what are the active ingredients, who are the target group, when should you apply the intervention, and would you be combining this with an additional adjuvant treatment, whether it's a non-invasive brain stimulation or a drug or some other treatment. And then we want people to think about whether there's a go or a no-go decision that should be made about whether you take it through into a large trial. I'll talk you through that in a little bit of detail. So this is sort of what it looks like, the knowledge units. Do we have the right evidence? Are we confident in the evidence? And then do we go into go or no-go? And careful consideration is what we want to do to optimise our chances of success. So if we think about the knowledge units, how much, when, who, the what, and whether there's something else that we apply as part of a treatment, because these are about, often our interventions are behavioural in nature, whether it's a speech and language intervention, whether it's a cognitive intervention, whether it's an intervention for motor function, sensory. Often we're talking about some kind of behavioural intervention at this point in time. So if we think about it, we want people to go, okay, here are the knowledge units that are important for us. Do we know enough, and how confident are we that we know what we should be putting into our trial? So we think about it along this continuum. We're not confident at all, so we're going to have a guess. We're somewhat confident because there's evidence, or we're very confident and we feel like we've got the target, we know what that is. So one of the other things that we've asked people to think about, and we've built up through consensus, are these important considerations for taking an intervention into a research study. And again, I don't want to put people off starting doing research. That's not what we're talking about here. We're really talking about when we want to build collaborative efforts to get studies done that we think are going to make a big difference to someone's recovery, or at least that's what the intervention precursors are telling us. So you'll see if you look on the right-hand side of the screen, it says prior research that's appropriately large and has been done shows us that we are getting changes that we would like to see in this population. That there's been a sequence of other studies that, again, give us confidence that we're moving from one piece of knowledge into a bigger piece of knowledge, and we bring that together into our trial. That we have some clear rationales that hopefully are mechanistically and behaviourally well conceptualised, even within the limits of what we don't know. And we want to think about whether or not this effort builds on other efforts that have been independently replicated. And the same for the preclinical. You know, we actually need to think about how we design preclinical research also. And there is actually quite significant parallels in our thinking about how those preclinical research studies might be done also. So coming back to the trials development framework, it would definitely be a go if when you looked at all of the evidence related to these key knowledge units, you felt really confident that you knew what it was you were targeting for that study. Then you can take that, you work with your methodologist and your trialists, and you build a trial based on knowledge that is well powered and able to be conducted accordingly. But if our frame at the moment, for whatever the issue is not confident, somewhat confident, and in reality, this is where we are for quite a lot of our knowledge at the moment in recovery, then that's a no-go zone. And we don't mean you don't want to do research here. We actually really want to do research here. It's just that we might not do the research in a randomised controlled trial fashion. Because if we decide to do a large randomised controlled trial without really knowing what all these elements need to be, then we're sort of having a guess. And that is something that we really want to avoid for a number of reasons. We'll come back to that. But what we also spent some time doing saying, well, if we don't have answers to those questions, how do we find out and what methods might be used? And this is where we touch on some of the newer designs, which are designs like adaptive trial designs, dose escalation designs, which can be quite helpful for trying to look at describing your intervention. And if you don't know how much you want to deliver and in whom, then you can do these smaller dose escalation designs in multiple cohorts of people, and you can find important dose limiting events or some efficacy signals that can give you more confidence that what you're going to be tackling is the right thing in your study. So these are new. We're really moving to these, and it's not simple because one of the problems that we have with some of these maybe smaller designs, earlier designs, is that a lot of our research networks and our funders are really favouring large randomised controlled trials. So they like to fund these big studies. And they're less inclined to fund these smaller, clever design studies. They don't. So we've got a bit of education to do about this as well to our funders. So we came up with a number of recommendations. We would really encourage people to try and use the trials development framework and to see if it's useful to them, and we really welcome feedback on it as well. We're encouraging people to not jump straight into a study when they don't have enough knowledge, and we've made a number of recommendations here that you can go to in the paper. I won't go through all of them now. And just to finish about whether our trials should be multi-centred, well, yes, they should, because Multi-centre studies help with our generalisability. They also help test feasibility for our studies beyond our own centre, where there might be specific things at play, and we really need to see whether or not we can deliver this intervention outside of our own centre. And of course, they help us build collaboration and give us more robust results. But we really want to take the best interventions through to the larger scale trials. And that's what we're targeting now as a group. And that is both to optimise our chances of breakthrough, but also to reduce waste in time and also taxpayers' dollars, because much of our research is funded by the taxpayer. So I just want to acknowledge my group. I have a wonderful group of people. I also have a fantastic network through the Stroke Recovery and Rehab group, which includes over 80 people from across the globe who've been working together over the last 5 years. And we're now taking, from the roundtable, we're moving forward with developing an international stroke recovery alliance. And if you're interested in being part of that, I'd really encourage you to sign up and join and help us build this alliance, because it's so early days. We're not sure exactly what it's going to look like, but we welcome you to participate in that. Thanks.
Thank you very much, Julie. That was absolutely wonderful. And so just for everyone's information, at the bottom of your screen, there's a Q&A panel where you can type in a question. We've also got a question off the chat. So before we go to the questions that have come in, thank you, Julie, firstly, for a wonderful talk, a reminder that we need to keep thinking about stroke in the middle of the COVID pandemic. We need to not neglect stroke. A couple of points I want to pick up on, which I think are really, really important. The whole question of thinking separately, that thinking about recovery isn't the same as thinking about rehabilitation. The two, and you need to try and separate them out. And I thought you did that really nicely. And I think the other point that you make, that rehabilitation trials are by and large funded by the taxpayer, and there is scope for huge waste if you go into a big trial, use up all your friends' energy and time and everything, and taxpayers' money. And so that's why I think setting out the steps that you need to take before you get to that, I think has been really helpful. And I think the other thing is that actually, if you take that rather more cautious approach, you're more likely to get a really good trial designed, more likely to end up with something that really will persuade the funders, and of course all of your colleagues around the world to really put their shoulder behind the effort.
Yeah.
So I'm going to take Chairman's privilege and ask a couple of questions, and then we'll open the floor to the questions we've also got. So the science of how to do rehab research is clear. Recovery and rehab research is moving forward very quickly. So do you have a sort of one or two top tips for folks that want to do research? What would be your sort of 2 or 3 key top tips?
I feel, and I've learned a lot, and I want to publicly acknowledge you as one of my really critical mentors, Peter, in doing trials. I've learned so much from talking to you over the years. But I do, as someone who came, when I first spoke to you, I was doing a small study, 2 centres, and I think the thing that you learn is if we can collaborate more, that would be a great thing. So learn from others. So if you want to do research, I fully encourage you to do that. I would encourage you to do 2 things if you can. 1, join someone else's trial if it's a really good trial, and if you feel it's an important question. You can learn so much by just being involved as a centre in somebody else's study. So I think that's one thing that I would really encourage you to do. I acknowledge we don't have a great mechanism for helping to do that yet, so that's something I hope the Alliance does. The second thing, though, is if you are going to embark on research, both use this framework, but use other people. So make connections with people who know what they're doing and who have a lot of experience. And because in research, we are actually a very generous, especially in this area, we're a very generous community. And so just reach out to someone if you've got an idea and you want some advice, and no doubt they'll give you some time to talk about it. So those are the two things I would say: reach out to get knowledge and assistance, and join someone else's trial if you'd like to learn without all the responsibility of starting a study.
I've just got one more question. So it's a question about so in the world of stroke, around the world, there are lots of places where resources for stroke recovery, stroke rehabilitation, or stroke in general, are quite limited.
Yes.
And so when you're designing research, how do you think about making sure that you choose an intervention that could be implemented widely, not just in centres that have got lots of resources? You know, how can I make my intervention something that lots of people around the world could do? Do you have any tips on that?
Right now it's a really critical, it's showing up the real gaps we have in rehabilitation services and what we can deliver. So everyone who's interested in rehab right now will be trying to get onto telling anything to help people continue their rehabilitation after they go home. So there's certainly and tele-rehab has some real challenges in developing countries as well. So I think we're at a really exciting phase where we're both going to see acceleration in how we do our research. And to answer your question specifically, Peter, the only way you know whether or not a country might be capable is if you actually design it with them in mind and get them involved from the beginning. So that's the answer. If you want to try and make sure it's going to work in another place, then bring them in from the start, because you can only develop this stuff with people's input. So that's one thing I would definitely say. And then the other thing I would say is, and it's happening for me right now with the dose, our big adaptive trial that's happening, is that we're looking for opportunities to leverage research to help build practices that are helpful in a country. And that, again, that's a great thing that research can sometimes offer. You can leverage the research to help drive some kind of useful change. Again, though, it can't be done without you being right there with those clinicians at the coalface in that location to find out what they need.
Okay, so we've had some questions in on the Q&A button. So the first is from Rustam Salman in Edinburgh. And his question is, do you think failure is a neutral trial, as you suggested when discussing focus and RATOLs? Isn't failure just poor methodological conduct? And how often do you expect stroke rehab trials to identify a positive or beneficial effect?
Yeah, really, really good. I use that word in parentheses, Ruston, because that's what it's called in our world. People just go, it just failed. Of course, I agree with you. Both those trials that I gave as examples were well-conducted trials at a methodological level. Were they well-targeted trials? Certainly, I think that's where we're thinking. That's where we're starting to see this thinking. So it's really about how we optimise. And I would say, I think when we do these research, there's so much we learn. Every time we do them, we learn something. We learn something about how we do it, about how we measure it, about how we collect data, about the design. So I don't think they're failed, but that is how they're described. And I think our target, yes, of course, we'll have lots of failures or lots of neutral studies. What we need is a really rigorous approach to try and get the best interventions into trial, and that's our objective.
Okay, there's lots more questions, so I'm going to try and get through them. So,
and I'll be quicker.
Kari Thekian Gopinathan is asking, is there any research around tele-rehabilitation?
There is. Yes, there is. So there is some research around tele-rehabilitation. There is and as I said, everyone is scrambling right now to look at what is the right kind of model. Tele-rehabilitation or certainly teleconsultation is tested generally to look at whether or not it's as good as face-to-face delivery. So a lot of our trials are looking at whether or not whether it's superior, just whether it's equivalent. Okay. And there's been some studies of that. And Steve Kramer presented one a couple of years ago. But I just want to say that there are a number of people sending within country, sending out links of any available resources. So please contact me if you want me to send it out.
Okay, and I'll just remind everybody that there's the slides and the talk will be up on the World Stroke Organization website, and it will be you'll be able to get the links and the free papers of the International Journal of Stroke with lots of background information. Another question from an anonymous person: do you think the effectiveness of any randomized trial to some extent depends on the cultural constraints and the model of practice in that organization?
So this is where my comment about multi-centre trials comes into play. So I do feel that if you do a study in one setting, with one culture, with one group, that you may see a very different kind of result than if you broaden it out to another setting with another group and another population of people. Because even in my country, there's regional differences in the population. So, yes, I do think it can be the case that you can see something that's positive within one study, but when you try and broaden it out, that changes because of the variability that you start to see. Ultimately, for us, to have an effective treatment, we want to see it work in multiple different settings. And that's really important. And that's why I'm encouraging multi-centre studies.
Okay, and now there's a question dear to my heart. When designing studies, do you have any tips for participant engagement in longitudinal trials where we're asking people to come back up to 5 times for scans, tests, and question lasting 4+ hours? I mean, my answer to that is you don't want to be designing trials that require 4 hours of patient participation from the word go, but that's my perspective. So, Julie, what's your view?
No, I must say I agree with you. And look, we held we're really interested in how we work closely with stroke survivors to help them understand research and for us to help, for them to help us understand how we should be building our research. And I think when you talk to a consumer, one of the tips I would have for you is when you're designing your study, make sure you have a user, so a stroke survivor or several, part of that conversation from the beginning, because they will tell you, no, that's not on, or that's okay, but you need to tell us this. So at the design stage, do involve your consumers, your users, the people you want to be following up. They will give you good information. The second tip I would have for you is it's about communication. It's about trying to make sure that you cue people, what's happening next, you keep them on board. You might have tools that keep them engaged with your trial that just, touch base every now and then to help them feel connected. It will work with some people, but it won't work with everyone.
I think the point about working with people who will join the trial is very important. I'm involved in a long-term trial where we discover that we're only 30% of the participants are women. And so been trying to understand why is it that women don't want to join this long-term trial. And, we're in a sense, we've discovered this too late. We should have really tried to sort that out at the beginning so that we could have ensured appropriate numbers of proportion of females in the trial. The second thing is, and I'm involved in a trial testing treatments for COVID. This is a trial that got started randomizing within a week of getting the grant. And I think,
Fantastic.
but it's designed on being super lightweight. So I think the general rule is if you want to make a bigger, affordable trial, you have to go for really simplifying things. And this comes back to what Julie's been talking about. Work with the people that will be working with you on the study, work with the people that you might be recruiting to the trial, and then think very hard about cutting everything down to the absolute essential.
Yeah.
Okay. So another really good one, Jan Verbeek, I'd like to know your view regarding whether we need more observational recovery studies as recovery patterns and patient profiles after the introduction of thrombectomy do seem to have changed.
Yes, I agree. Yeah, I think that's a really good point. Clinicians, rehabilitation, recovery people are seeing this sort of bimodal distribution now where we've lost the middle group and that they're either all doing really well or they're quite severe cases. So I think we are seeing some really interesting stuff. If that's real, that's the comments we're getting. So, but you're right.
Questions, Julie.
No.
Got to keep rolling in. So,
okay, sorry. Yes, we do need more observational studies. Careful ones.
Yeah.
And we measure along the way.
Yeah. Okay. So Jonas Pols asked two questions. Can we find the Holy Grail? What's the chance we've missed the efficacy of interventions on restitution?
I think I'm an optimist. So I think we can find treatments that are going to markedly change recovery. I think we're moving towards that. We're learning a lot all the time. There's some great interventions, not just behavioural ones. Might we have missed something? Possibly. But I think with our knowledge, we might end up knowing that target and go back again and pull those back out.
Okay, so now a question about translational research from Daniela Garcia. What do you think are the most prominent challenges in the translation of preclinical to clinical research when looking at plasticity?
So I think when we did the first roundtable and looked at translation between preclinical and clinical, it became really clear that we're not really talking enough, and the types of designs that are being used in some of the preclinical research is not necessarily giving us the kind of information we need for translating across into clinical. So there's actually some great recommendations from the translation paper that I would recommend. And also in this trials development framework, the preclinical scientists provided evidence, and they also talk about where the gaps are in evidence and how they need to be filled. So I'd recommend you having a look at that.
Okay, so some papers to go away and look at. Another question about methods, which is rehabilitation trials that look at processes of care and services require different methods to trials looking at recovery. Can you tell us your thoughts about that?
Yes, they do. Quite right. And so rehabilitation trials where you're trying to implement something into a service requires a different kind of design, and there are a range of different methodologies. We don't really tackle that in this paper because it wasn't the target of the discussion. There are actually some good papers out there, and I'm happy to pull some up and put them on this webinar later.
Great. Okay. Another question about stroke patients often suffer cognitive impairments, losing decision control. How do we take these factors and the degree of motivation into account when designing clinical trials?
Well, we did cognition as a topic for the last roundtable because we know it's so complex. I think that there are strata that we might need to consider. So we might need to stratify our studies by someone's cognitive capability, their either understanding. And also, can I say, I think it's really important we include people with aphasia in our trials, especially our trials of motor disorders. I don't understand why we can't put them in, more about the outcomes than not being suitable. So aphasia we need to include. And then the other point I was going to make, so the cognition stratification, motivation I have a problem with. I don't know if we know how to measure motivation. I've been struggling with this for decades. And but I do think we can measure cognition, and I think we need to consider it very carefully and maybe stratify.
I'd like your point about the aphasia, because, of course, we all get hooked up. I mean, lots of ethics committees get tied up with this whole process of, capacity consent and, need to be able to sign a consent form or give verbal consent. And actually, that is a barrier to evaluating treatments for patients that can't speak or give consent. And so I just say it is possible to do research, and we just need to work harder with our the people who we're going to do research on and the ethics committees to say, look, we can now do emergency research in people who are so sick they can't give consent immediately. Surely we can manage this in rehab research with people who are cognitively impaired or with speech disorder.
And our position is that it's both unethical, because why should they miss out on being able to be involved in trials, but also that they make up a big proportion of patients. If we don't include them, then the results will never reflect their problem.
I mean, Richard Lindley did a really nice study back when we were trying to do a study of thrombolysis in elderly people. And lots of people said, oh, you can't do elderly people. They're far too high risk. No, they shouldn't. So Richard asked lots of elderly people, how would you feel? Trading off the risk of dying from the treatment or maybe surviving with less disability. And they said, well, go for it. And so that's what we put in our informed consent. So I think working with patients is or people who might participate is really important. Okay, yeah. Another great question from Louise Connell. I agree that we need to think beyond multi-centre RCTs, but how do we convince funders of this?
Right, so that's right, Louise, and we recognised this. So in the paper, one of the things we did when we summarised the evidence and said these are the targets, is we need to socialise these targets, and this is just an example, and we asked the funders if they would write a commentary. Now, we only got a couple of funders that agreed. The UK funders did, Canadian funders did, and the Australian funders agreed to write a commentary about why they thought it was important to take on board the recommendations that come out from the community. It's an education piece. But also, can I say, there's got to be an excitement piece in here too. The way we'll get funders excited is if we start to show them something that looks exciting, and then we can convince them that the quality of the science is so good that they can't not fund it. So I think our major barrier is in these earlier studies that we need to really show the evidence that we can then pull in.
This is a controversial question from me. Do you think that funding panels are biased by basic science researchers who do small experiments on small numbers of animals and small numbers of test tubes and can get answers in a few weeks, and they're just not used to thinking of our kind of longer term, play the long game kind of approach to research? Do you think there's a basic science bias?
Well, I don't want to. I do work at an institute full of basic scientists, so I have to be somewhat careful. I do feel that we don't understand each other's fields, and that can create a problem. And I'll also say as well, Peter, that, I've encountered physicians who are used to doing mega trials that don't understand what recovery trials are trying to do either. So I think it's about us trying to educate people and get them excited by our science.
Absolutely.
And the reason we need to do this is because if we keep not having successes, then people get bored and disinterested, and they label the whole field as being useless. And that's what we're seeing now. We've got to get over that.
Okay. So we've got another one. Should we not just focus on smaller studies in the future if the results are positive with these trials? So,
right. So if we do really well conducted trials, and if the trial shows benefit, and that would be amazing, because then we would have to have something that is very powerful to show benefit in a small study. We still need to do more centres because we've still got to replicate to make people feel confident that these these results are real and robust. So although I do think it would be so exciting to see the intervention so powerful that we can show an effect in small studies, we wouldn't necessarily need to do them in thousands of people, but we would still need to replicate them to be able to convince everybody that this is working and it could be applied in different settings.
I mean, the FLAME trial is a very good example. FLAME trial in France suggested fluoxetine was positive, helped motor recovery, very exciting. And, the large-scale trial FOCUS hasn't replicated it yet, although there's 2 more trials on the way.
Yes, there are.
So I think that's an important point that, small studies do have a value. They get people excited. But sometimes small studies may be falsely negative or falsely positive, and that's why you need to replicate. So from Yuri, we've got 2 questions. Firstly, are the stroke phases that you mentioned on your first slide evidence-based, taking into account the heterogeneity of stroke? Are the phases all the same, if you like, in different, lacunar strokes, large strokes, or is that just a kind of overall average?
You mean phasing, like phase 1, phase 2, phase 3?
I think he meant the phase of hyperacute, acute.
Oh, sorry, the timing.
How the recovery process operates in different subtypes of stroke.
No, I don't think it's exactly the same. So we see, of course, in intracerebral hemorrhage that the recovery profiles of patients are very different. And we see in aphasia, in fact, that the timing of recovery is different to motor recovery. Yeah. So different subsystems and different particular biological issues are at play here. One of our big problems, which takes me back to Jana Verbeek's question around observational studies, is we still don't understand recovery phenotypes very well, and that's another major area of research that we have to really get to grips.
Okay, Jenny, you're doing fantastically, Julie. I'm afraid the questions are still coming in, so if you're okay to keep going.
Yeah, sure.
Okay, so Yuri asked another question. How can we propose our centre for participation in studies?
Yes, so that does bring me back to this issue of what is a good mechanism for us at an international scale to try and bring people together. We've got the Global Alliance of International Stroke Networks. They do have some interest in rehabilitation and recovery studies, but it's still, not in every country are there these trials networks. If you have a trials network in your country, I would recommend you join it. Even if right now it's targeted at thrombolysis or thrombectomy and acute drugs, join it if you're a rehab or recovery person, because you'll learn and there'll be other people that might join it as well. So I'd definitely do that. Second thing is please join our alliance, because I think we're going to work towards developing flagship projects and we'll be looking for centres.
Okay, so now we've got Nick Kudierski asking, how important is it to consider the influence of the patient-practitioner interaction, communication techniques used to recruit, engage, and motivate participants? So how do you handle the, because being a therapist or a rehabilitation practitioner, that interaction is a pretty key part of the chemistry of the intervention, if you like.
Yeah, that's a really great question. I actually got asked years ago whether or not I did personality testing on my therapists that were involved in my trials. And I thought, wow, that's actually an interesting question. The reality is I do think there is a relationship. In our trials, there are lots of different people delivering interventions to lots of different patients. And of course, that will vary even with the one individual therapist. How I gel with a patient will be quite different from patient A to patient Z. So although it's a really interesting area, it washes out when we do these bigger studies because this variability will be present all the time. And so I do think it's a really interesting question. I think it's an interesting question for when you're, especially if you're doing rehabilitation research and you're trying to roll something into the clinic. I think the therapist attitude or the nursing attitude or the doctor's attitude to that rollout is absolutely critical, and I think you need to capture that. I think less so perhaps in the recovery study. Good question.
Okay, so Jan Verbeek has got 2 questions, and mindful of the time, I'll go with one of them.
That's my dog, sorry if you hear that noise. He's a geriatric dog. You can feel sad for my dog.
Oh, poor chap.
That's okay.
He's got a cough. It sounds like he hasn't got COVID, I hope.
No, he does not have COVID.
Good. Okay. So what's your view on using data collected in routine clinical practice for answering scientific research questions?
So I work with a fantastic statistician, and he has this statement. You can have good data and you can have big data, but rarely do you get good big data. So although I welcome routinely collected data that's collected in a really standardized way, say in a registry, I can see how there's registry-based data and we might be able to add modules into the registries that are well collected. I'm a bit more sceptical that routinely collected data is of high enough quality to give us the insights that we need, unless it's a huge data set, but even then I feel like you would have to confirm it some other way.
So in a sense, I guess, is it fair to say that big data can tell us about the process of care, can tell how care works in your health system, but it doesn't tell you what the effects of that care are, whether it's doing, is it better than another form of care? And if there's variation in care between sites, that just tells you there is variation in care, but it doesn't tell you anything beyond that. I think that's a big.
Yeah. And also I think, though, it can be hypothesis generating, right? It could help you generate hypotheses about particular subgroups or about people who have some kind of predisposition or pre-existing problem. So we can generate hypotheses, but I'm not sure it helps us answer.
Okay, I think we've still got a couple more questions, but I think, Julie, we've worked you very hard. You've done a fantastic job. Thank you. And so we've got some thanks here from Yuri. Thanks for a wonderful webinar. We much enjoyed it. I'm sure there'll be lots of other really positive comments. And of course, people, don't forget, you can come back and listen to this all again. We had over 80 participants, so fantastic success. And thanks to the World Stroke Organization for organizing this. Thanks to Alina for handling all the technical complexities. And so on that note, I'll say thank you very much, Julie. We'll let you get to bed because it's getting late now in your part of the world.
Yes.
And I hope your geriatric dog is comfortable.
That's great. Thank you very much. It's been a pleasure, and please keep in touch, everybody.
Right. Thank you very much.